Welcome to episode 8 of the Physiology 225 podcast. I'm your host, Dr. Jim Davis, and I'm excited to guide you through today's topic, membrane potential. This concept might sound a bit complex, but we'll keep it simple and approachable while adding just enough detail to help you understand its importance. By the end of this episode, you'll have a solid grasp of the basics. So let's dive in. To start off, what is membrane potential? At its very core, membrane potential refers to the difference in electrical charge between the inside and the outside of a cell. It's like comparing two sides of a battery. One side is more negative, the other more positive. This difference in charge is crucial for many cellular functions, particularly in excitable cells like neurons and muscle fibers. While this explanation gives us the gist, determining membrane potential involves more intricate interactions among ions, gradients, and membrane properties that we won't get into in this class. How is this membrane potential established? Well, this membrane potential arises from the distribution and movements of ions like sodium, potassium, and chloride, as well as the selective permeability of the cell membrane. So let's break it down step by step. First, we have ion concentration gradients. We're going to focus on sodium and potassium. I mentioned chloride a minute ago. It's important, but not as important as these other two. Now, sodium ions are more concentrated outside the cell, while potassium ions are more concentrated inside. This uneven distribution is maintained by active transport systems and is fundamental to establishing membrane potential. Next, we have leak channels. Potassium leak channels allow potassium ions to move out of the cell more readily than sodium ions can move in. And this is due in large part to the difference in concentration gradient, as well as the fact that there are many more potassium leak channels than sodium leak channels. So in other words, a way to look at this is there's less resistance to potassium flow than there is to sodium flow. Now, because potassium is positively charged, this outward movement makes the inside of the cell more negative. Next, we have negatively charged proteins. Large negatively charged proteins inside the cell that are close to the membrane cannot cross the membrane, which adds to the overall negative charge within the cell. To be clear, there are positively charged proteins throughout the cell, but the ones that are nearest the membrane are generally negatively charged. Finally, we have the sodium-potassium ATPase pump. This vital active transport mechanism moves 3 sodium ions out of the cell and brings 2 potassium ions in, and it needs ATP as an energy source. By pumping out more positive ions than it brings in, the pump helps maintain the negative resting membrane potential. That's really its job. The job of the pump is to maintain negative resting potential. Without the pump, we would fall into equilibrium, and as we've talked about before, equilibrium in physiology usually results in death. Now, the result of these interactions is a resting membrane potential of approximately negative 70 millivolts in most neurons, meaning the inside of the cell is 70 millivolts more negative than the outside. Coming back to the pump, the sodium-potassium ATPase pump is more than just a transporter. It's a key player in maintaining the cell's electrical stability. By continually working to offset the effects of ion leakage, the pump does a few things. It keeps sodium and potassium concentration gradients intact. It prevents the membrane potential from reaching equilibrium, which would halt essential cellular processes, #death, and it also helps the cell stay ready to generate action potentials when needed. And we'll talk about action potentials in a couple of episodes. This constant activity requires ATP, highlighting the pump's role as an energy-intensive but vital mechanism for life. I think I read somewhere that they estimate that up to 40% of the energy used by your brain goes to powering these pumps. So you probably have a question, why does membrane potential matter? Well, it's a fundamental, membrane potential is fundamental to the way excitable cells like neurons and muscle fibers function. So here are some reasons why it's so important. Action potentials. The membrane potential sets the stage for rapid electrical signals, enabling communication across the nervous system and triggering muscle contractions. Secondary active transport. The gradients established by the membrane potential drive the transport of other molecules, such as glucose or calcium, into or out of the cell. And finally, and perhaps most importantly, homeostasis. Membrane potential helps regulate ion balance and cellular activity, which ensures cells function optimally. So let's summarize with our key takeaways. Membrane potential is the charge difference between the inside and outside of a cell. Typically, it's around negative 70 millivolts in neurons. It's established by ion gradients, selective membrane permeability, and active transport via the sodium-potassium ATPase pump. The sodium ATPase pump, while establishing the membrane potential, plays a more important role by maintaining that membrane potential. And it does this by maintaining the gradients of sodium and potassium, which prevents equilibrium. It needs to use ATP to make sure that the cell's is electrically and functionally stable. Membrane potential is essential for processes like nerve impulses, muscle contraction, and secondary active transport. And with that, we're done with episode 8. I hope this expanded explanation of membrane potential has made the concept clearer and more relatable. Join me next time for episode 9, where we'll begin our journey into the nervous system, focusing on its primary functions and different types of channels. Until then, keep questioning, keep exploring, and stay curious.